I'm Bob Wachter. I'm chair of the Department of Medicine. And today is a special day, both because of our guest lecturer, who appears to know a lot of people here, because I've seen a lot of hugs going on in the front row, but also because it's the Rudi Schmid Memorial Lecture. And Rudi was really a central figure in the history of UCSF and of our department. And I'll ask Gabriel Ma, a professor of gastroenterology, to come up and say a few words about Rudi before we introduce the speaker.
Thanks, Bob, and thanks everyone for coming, both of you, all of you who are here, and as well as those who are online. I think I've got a lot of requests before this meeting about the Zoom links, so I know that there's no order of magnitude, I think, in the virtual space here. As Bob mentioned, we're here to honor the legacy of Rudi Schmid. For those of you who don't know about that legacy, Rudi was the inaugural founder of the GI division here, and really started the tradition of bringing science to medicine. He was actually a hematologist by training, and studying porphyrias, he realized that part of the mechanism of the disease related to liver enzymes. So he transitioned quickly to understanding the biology of the liver, and that was really his entree into the field of gastroenterology. It had nothing to do with clinical gastroenterology. It really came from understanding the science and wanting to understand the mechanisms of disease. And so Rudi both founded and led the division for many years, and did such a wonderful job that he was then asked to be dean, which he also led in that regard. UCSF did many wonderful things for us. But we're really honored today that we're going to have someone as notable as Charlie Rice to continue honoring Rudi's legacy. So welcome and thanks. And Bob will tell us.
Great, thank you. How many do we have online, Alison? Yeah, so we'll have a few hundred people, and that they will come on. Really thrilled to introduce Professor Charles Rice, who's the Maurice and Corinne Greenberg Professor and Head of the Laboratory of Virology and Infectious Diseases at the Rockefeller University. He's a leading, you might argue, the leading expert on flaviviruses, including hepatitis C. He's authored over 500 publications. He asked me to shorten the number of honors, so I will, to make sure he's got time, but we have to go through a couple of the big ones. Past president of the American Society of Virology, fellow of the American Association for Advancement of Sciences, member of the National Academy of both Sciences and National Academy of Medicine. Also earned a share of the Lasker-DeBakey Clinical Medicine Research Award in 2016. And in 2020, he was awarded the Nobel Prize in Physiology or Medicine. So we are honored to have him here to talk to us about a cure for hepatitis C. Now what? Thank you.
So thank you, Averill and Bob. It's a real sort of pleasure to be here. We got some people sneaking in the back. You guys can come down to the front here. There's plenty of room. Don't be shy. The food is closer out there too. So, okay. So today I'm going to give you kind of a bit of personal history of sort of my sort of chance encounters that led to some work on hepatitis C. I don't have anything to disclose. So I'll start with the trials and tribulations of tackling hepatitis C, which turned out to be, sort of quite a frustrating time for a molecular virologist. Talk a little bit about sort of what we have now in terms of being able to treat this disease. And then I'll end with a new model that I'm very excited about, which I think fills a gap in our ability to understand Hepativirus pathogenesis in terms of progressive liver disease and cancer. And a few concluding remarks, if I have time. All right. So a little bit about, sort of how I ended up, sort of being interested in viruses, which is this is 1975, sort of many of you were not born at that time, I realize that. And I wanted to go to graduate school, and I was interested in developmental biology, and in fact, sea urchins. There was a really good lab at Caltech that sort of did sea urchin development. I'd worked on that as an undergraduate. When I got there, I actually got placed in a virology lab in the Strauss lab. That was my first exposure to viruses. And I really just sort of enjoyed the ability to work with these entities where you could, sort of take a virus, sort of add it to a cell, and you had a very defined perturbation that you could study. So I began in Jim's lab studying RNA viruses. And this was kind of a special time that's not dissimilar from the one that I think we find ourselves now with the sort of CRISPR revolution. It was the mid-70s, sort of the dawn of molecular biology. We could finally actually do nucleic acid sequencing. It was a lot different than sort of dropping your envelope off and getting the sequencing back the next day. But nonetheless, we could begin to understand and unravel that. Reverse transcriptase had been sort of discovered by Baltimore and Temin, so you could take RNA and convert it into cDNA. And was sort of lucky in the sense that the Strauss lab at Caltech was just sort of down the hallway from Tom Maniatis' lab. And Tom Maniatis was sort of one of the major players in recombinant DNA technology. And he had a postdoc, Eugene Butler, in the lab, who studied bacteriophage DNA-dependent RNA polymerases, which you guys probably know now as sp6 T7 polymerase. And this allowed you, knowing that promoter and having purified enzyme, to basically make unlimited quantities of whatever RNA you wanted to make in a test tube. So if you sort of think about that and an interest in RNA viruses, in particular positive strand RNA viruses, this was an obvious sort of menu for being able to actually sort of manipulate viral genomes and test structure, function, make mutants, and see how they behave. So, classically at this time, a lot of the work that had been done was, forward genetics. You sort of mutagenize your viral population, look for a mutant that has an interesting phenotype, but you wouldn't necessarily know what genetic perturbation had actually changed that. And reverse genetics is really, sort of what we wanted to do. In other words, sort of know the sequence, maybe get interested in a gene or one of the viral proteins, make mutations and study their phenotype, reverse genetics. And so this was a major effort in, I think, all of the labs sort of studying RNA viruses. And it had been demonstrated in a bacterial virus system from Charles Weissman's lab that, basically you could take a cDNA copy of an RNA virus, transfect it into a permissive bacterium, and it would sort of launch that RNA virus and it would grow. So this is what we were trying to do in the animal virus space, sort of in the 70s. And Vincent Rockefeller, who some of you may know, a really famous poliovirologist who worked with David Baltimore, was the one who really sort of got this going. And it was interesting because I think we believed that actually expressing these viral RNAs precisely was going to be important for their functional activity. But what Vincent did was basically just take a full-length cDNA copy of poliovirus in the sort of garden variety plasmid that was used at the time, PBR 322, I think, and transfect it into polio-permissive cells, and voila, he got virus. So this was kind of a Hail Mary experiment that, sort of was the first example of taking a cDNA copy of an RNA virus, an animal RNA virus, and showing that that could give rise to a complete virus sort of infection cycle. So at about this time in 1986, I was taking my first faculty position actually after a short break in Canberra, Australia. And, when you take a position, you're supposed to write down what you're, thinking of doing, hoping to attract some, fantastic students and postdocs. And so this was basically what I had in mind. Given the capability to introduce point mutations, deletions, and insert foreign sequences into these viral genomes, you can test structure-function relationships. In addition, opens up new avenues for the design and propagation as recombinant DNA of live vaccine strains. We were particularly interested in yellow fever in that regard, and should allow us to explore possible use of these viruses as vectors for the cytoplasmic expression of foreign genes in animal cells. And I should say that, I think a lot of virologists think that their particular virus that they're in love with can do everything. So this was kind of like a reflection of that. And but that's kind of what we did. Sort of when I got to WashU, we for Synbis virus, which is an alpha virus related to chikungunya, which you guys may have heard about. We made a functional clone for Synbis using SP6 to generate RNA from cDNA and showed that it was useful for mapping mutations and temperature-sensitive mutants and basically doing in vitro mutagenesis to develop mutants. We also showed that you could actually make a subgenomic replicon of Synbis virus, which was useful for gene expression. And this is, some of you may now, we're in an RNA world, although we're not supposed to talk about RNA or messenger RNA. But this replicon system actually was what are now called self-amplifying RNAs. This is, I think, to sort of delete the fact that they're derived from an alpha virus, usually Synbis or Venezuelan equine encephalitis virus, which doesn't probably sound too great if you're trying to sell a product. Anyway, it took another 30 years or so for this to catch on. But we had also been busy with another virus, yellow fever, which was a prototype Flavivirus. And at that time, there was really no information on what the genome structure of this virus was. There had been a lot of early work done, but it was a prototype member of the family. I think many of you know about sort of yellow fever as being, sort of the virus discovered in 1900 and shown to be sort of mosquito transmissible. I think they're doing some construction up there. And the other thing that is, I think, notable about yellow fever is, in fact, that there was this vaccine strain derived by Max Tyler by passaging the virus in moused up tissue. and Max sort of won the Nobel Prize for that in 1951. It's a vaccine that's been in about 500 million people, so pretty interesting to sort of know. And it's kind of the ideal vaccine in terms of one shot, and we believe lifelong protection against even diverse infections with wild yellow fever isolates. So we determined the sequence of the vaccine strain as well as the CB strain, which was the one that Max started with. And as for Synbis, we tried to sort of make a functional genetic system to begin to study yellow fever, to map the changes that were important for attenuation and immunogenicity. And unlike Synbis virus, it turned out to be really a pain to do this because the full-length clones of flaviviruses are toxic for bacterial strains. And so finally, after years of frustration, we just decided, well, we're not going to really sort of get a plasmid that has a full-length template of HCV, of yellow fever. Let's just go ahead and take 2 restriction fragments, ligate them together, use that as a template for transcription, and that finally worked. So, you could imagine that in today's world that these 2 viruses that I was working on, Synbis virus and yellow fever, would fall into the category of, why are you doing this? Synbis isn't a human pathogen of real great sort of importance. And I think that the other thing, and even my mom brought this up, says, why are you working on yellow fever virus? We have a vaccine for that, don't we? But at least at that time, it was possible to do things that you were curious about, where some of these systems that we were studying had advantages in terms of their ability to. So in parallel, what was going on was the description of an agent of non-A, non-B hepatitis. And this was recognized by Harvey Alter and also by Fred Prince at the New York Blood Center, who were seeing what happened to patients that were transfused with blood. And basically what they found in the mid-1970s already was that there were cases of acute and chronic hepatitis in transfused patients that were not due to the 2 known hepatitis viruses at the time, that you could actually be diagnosed hepatitis A and hepatitis B. And this observation, even in these early days, was important because it showed that whatever this agent or agents was could lead to chronic liver disease and cirrhosis. for example. And I think the other thing that happened was that it was recognized that going after figuring out what the agent was really important. And so this instigated a lot of effort sort of in biotech and pharma to identify what this virus was. Now, today, you guys would, take some clinical samples and, send them off for sequencing, and we know the answer in, sort of a day or two as to what the pathogen or pathogens might be. But in those days, that really wasn't possible to do. And this actually dragged on for nearly 15 years, sort of this quest to identify the agent or agents. And I think the real landmark was in 1989, when Michael Houghton and his group at Chiron, not too far away from here, sort of described this agent of non-A, non-B hepatitis. And this was accomplished by doing an immunoscreening protocol with convalescent non-A, non-B hepatitis serum, and then a cDNA library that had been made from the virus amplified in chimpanzees, which was the only animal host that had been found for hepatitis C at the time. And they got, one little spot that lit up with this, and it was a little piece of what then turned out to be sort of the HCV genome. And this was, I had been sort of at WashU for a couple of years when that happened. And basically, what caught my attention was that, hepatitis C was a lot like this virus that we've been studying, sort of yellow fever virus. So it's an RNA molecule, the genome, positive stranded, so can function as a messenger RNA, and similar to the Togaviridae or Flaviviridae. And then it says this molecular approach, which is the immunoscreening protocol, could be useful for identifying other infectious agents. So with that sort of discovery, it became possible to, sort of look around, and there really was, sort of one virus type that was responsible for this. It was distributed throughout the world. The initial estimates of the number of people infected were about 100 million. Various areas where it was particularly high, like in Egypt, that I'll mention later on. And this RNA virus, with its genome of about 9,600 bases, was really remarkable in the sense that it could establish chronic infection in the majority of people that were infected, regardless of their immune status. So about 70% of infected people after 6 months are still viremic. They haven't, figured out how to resolve the infection. And this smolders on to cause chronic liver inflammation, can develop into cirrhosis and hepatocellular carcinoma or end-stage liver disease. This is sort of a slow progressive disease, so maybe it didn't, receive the kind of attention that if it, virus is going to kill you acutely but nonetheless a huge disease burden. So in terms of thinking about working on this virus, it wasn't, sort of an easy sell. First of all, the virus had never been shown to be successfully replicated in cell culture. And people had tried this for years with the non-A, non-B agent, sort of using the chimpanzee infection model as a readout, and no luck. There was no small animal model, sort of only chimpanzees. So that's really sort of not a very convenient model, although we did learn a lot about the virus sort of from that model in the early days, which we don't use anymore for studies. And, I was confronted with, sort of, should we really start working on this virus? Because we had basically SINBIS and yellow fever. We had genetic systems. We could do everything that we sort of dreamed of doing with these viruses. And here was a virus that didn't replicate in cell culture. And so there were a couple of people that were crazy that did this, and that's actually me a few years ago on the left, and then Arash Grakui, who is the first technician I hired at Washington University, who turns out to have gone on and in his PhD done some things like discovering the immunological synapse, and he's now a professor at Emory University. But none of those things were apparent when I hired him. So I think one of the things that pushed us into doing something was this call from Steve Feinstein. Now, Steve Feinstein was at the FDA, but he had actually sort of trained as a fellow at NIH in the laboratory of Bob Purcell, who was sort of a great sort of player in viral hepatitis and liver disease. And Steve had actually been also trying to sort of identify this non-A, non-B agent. He'd also shown that, for example, if you took the agent and treated it with organic solvents, that inactivated it. So that sort of showed that it was an envelope virus. Anyway, I didn't know Steve, and he called up on the lab phone and said that, told people that he wanted to talk to me. And, what Steve had done, he's actually seen this paper on the yellow fever infectious clone, which was published in this obscure journal called The New Biologist that basically went extinct about, a year or two after we published the paper. So if you have a preprint of that, maybe you can get something on eBay. But he was interested in, could you actually sort of transplant hepatitis C envelope into yellow fever 17D, the vaccine strain, and thereby sort of derive a new vaccine for hepatitis C virus and maybe also for yellow fever at the same time. And we got to talking about this, and we sort of really had no idea what to do, because this virus, basically has a fairly long 5' non-translated region, and then this long open reading frame that gets chopped up by host and cellular proteases to produce the 10 proteins that the virus needs in order to replicate and make new virus. And we had no idea, sort of where these proteins really, the sort of mature proteins, started and stopped. So it wasn't like we could, just say, oh, this is the way we're going to make the construct. So I think this is kind of what pushed us into, sort of working on hepatitis C, kind of in a quiet way. Nobody else in the lab besides Rockefeller and I wanted to work on this virus, because they came to the lab to learn how to be virologists, and they didn't really have a virus that worked. So this is the sort of genome, as I mentioned, this internal ribosome entry site and long open reading frame. And Roche, really painstakingly made, sort of antibodies to different regions of the polyprotein, along with other groups around the world, and sort of figured out that the 10 proteins were generated by host and viral proteases. And people began to focus that were interested in coming up with better treatments for hepatitis C, sort of on the enzymes that the virus encoded that we believed were going to be essential for its ability to replicate. And this is the one of those combined enzymes, the serine protease helicase, NS3-4A, and then also NS5B. So I think one could use this information to begin to sort of develop biochemical assays for these, to sort of screen for compounds that inhibited them, but there was actually no way to actually figure out, are these really essential for virus replication? And will these compounds that we're developing through sort of biochemical assays going to be able to get into cells and actually block virus replication, because we had no replication system. So we went back to our sort of book of, trying to make a functional clone for hepatitis C, to develop a genetic system and hopefully figure out ways to get this to propagate in cell culture. And the material that we began with is actually from one of Harvey Alter's transfusion patients, patient H. And the reason that we were interested in that was because it was actually from the acute phase of infection, and its specific infectivity had been measured in the chimpanzee model. So we knew that there were sort of a lot of functional HCV genomes in this. So it made sense to start with it. The problem was that there was very little of this material that we had access to. So, we went ahead and sort of put a clone together, somewhat painstakingly with this prototype clone, and sort of sequenced it. And then the question was, I just told you we don't have a cell culture system to infect with this virus or transfect with this RNA. But we thought that maybe the block is at the receptor binding level. So if we bypass that by transfecting in naked RNA, it might sort of initiate replication. That was unfortunately not the case. So we're left with sort of the only other model for hepatitis C, and that is the chimpanzee. So how do you go from, sort of a transcribed RNA in a test tube to, being able to sort of initiate a liver infection in a chimpanzee? That's crazy. I mean, I think most of you that have worked with RNA, you kind of sneeze and, it's all degraded. So even if you're careful, it's not easy, but injecting it into the liver, sort of a naked RNA, yeah, kind of crazy. The good news was that actually this had been successfully done by Sue Emerson and Bob Purcell's lab, not with HCV, but with HAV, by injecting transcribed RNA into the liver of a marmoset and generating a hepatitis A infection. So it could work. So we tried it. It didn't work. So that was really pretty disappointing. And one of the things that you're going to sort of hear about in this talk is, occasionally we think we've, sort of gotten to the finish line on this, and then we find out that, nope, not there yet. This sometimes happens quite a bit with science. So we started worrying about whether or not the sequence that had been published by Chiron and the one that we had determined for the patient H strain was really the complete sequence. And that was because the initial report from Chiron said that it terminated with the 3' poly A track, kind of like most cellular messenger RNAs. And groups in Japan that were sequencing some other subtype isolates found a poly-U polypyrimidine track. But we knew from yellow fever that it didn't end in either of these. And in fact, there was this very sort of predicted stem loop structure at the 3' end of the yellow fever 17D genome RNA. And as you might guess, these viruses that only have about 10,000 bases to do what they do, don't carry a lot of excess baggage. So, if we didn't have the right 3' end, chances are it's going to be dead as a doornail. And we thought that was the reason that that had been the case. So I had a really fantastic molecular biologist who immigrated to the United States from Russia, Sasha Kolokolov, shown here actually quite a few years later from this actual event. And so he took on this task with, basically vanishingly small amounts of this patient H serum and actually trying to find ways to successfully clone and identify the sort of the 3' end of the genome. And I won't go through all the details, but in those days, that was, just sort of having a 3' race kit wasn't available. This was actually pretty painstaking troubleshooting work to do. And basically what Sasha found, and also what was found in Kunitata Shimatono's lab in Japan, was that there was a sequence of about 100 bases missing at the 3' end of the HCV genome. So if you look at this, there's a stop codon of this long open reading frame, a hypervariable region, an internal poly-U polypyrimidine tract, and then this sequence of 98 bases that turned out to be very highly conserved among all of the HCV isolates where it had been determined. So, we were pretty sure that this was the problem, So obviously what you do, you sort of plop it on the clone and sort of go back and do what we did before, try it out in cell culture and in chimps, and failed again. So, so this was, 5 years, 5-6 years after the discovery of this virus. We still didn't have an in vitro system to study it. Terrible. Fortunately, we did have some other things that were working in the lab, so we continue to get some level of funding. And I think that the other sort of virologists on the study section panels, sort of cut us a little bit of slack. It obviously wasn't at the same sophistication that people could do with poliovirus or whatever. But anyway, I guess we persisted barely. So, what could be wrong? Inefficient RNA delivery. I've already told you that, injecting naked RNA into a chimp liver, this would not be the way that you would want to transduce hepatocytes. Could there still be something missing? We didn't think so. We worked pretty hard on both ends of the genome. And maybe unlike, sort of the prototype for positive strand RNA viruses that, what if a naked RNA isn't infectious and you need some other factor, an RNA modification or a protein that's, sort of packaged with the virus particle? one thing's, these would all be, very difficult to sort of test and investigate with vanishingly small amounts of material. And then, sort of having the sort of chimpanzee model as the only readout. So one of the things that we could, sort of tackle were error-prone RNA replication. I mean, I think all of you know that these RNA viruses make a lot of mistakes. That's part of their secret of success, is to generate populations with a lot of variants. And but some of these are going to be lethal mutations. And maybe we had just incorporated some of these into our prototype cloning. And then in those days, the fidelity of the enzymes used for cDNA synthesis and cloning and amplification were not nearly as good as they are today. So what we decided to do was actually sort of sequence multiple independent clones across the hepatitis C genome from this patient age material, and then actually assemble a consensus sequence where at every nucleotide in the genome, it was basically the majority nucleotide. Now, it might mean that, that basically could have, that such a thing actually didn't exist in nature. But we thought that that might be a way that, would help us to sort of filter out errors. And in fact, it actually worked. And we I think we had sort of 10 variants that we tested in this experiment, because we didn't really know, for instance, how long does this poly-U, poly-UC track need to be? So we had a short version and a long version. We also had some clones that had extra nucleotides at the 5' end, so we made those. And we put in what we believe to be silent markers that would allow us to sort of figure out, what was replicating. Anyway, what you can see here are these two chimpanzees. And I have to say that our ability to do these kinds of experiments in the chimpanzee model was totally dependent upon Steve, because Steve Feinstein actually still had access to sort of the NIH sort of chimp facility, and Bob Purcell was very generous about sort of letting us use these animals for these experiments, because as a sort of an assistant professor, whenever in these days, if you infected a chimpanzee with non-A, non-B hepatitis or hepatitis C, the cost was to initially just endow that animal. And that endowment cost was about $84,000, $86,000. So for one animal, and not counting, housing and all the other stuff. So Steve was really, really key in this. So anyway, what you can see here in these two animals in blue is, for the first time, sort of rising levels of HCV RNA in circulation in these two animals. And then in red, ALT, which is a marker of liver damage, that sort of kicks in at about 2 to 3 months when the adaptive immune response recognizes something bad is going on and begins to sort of react and attack infected hepatocytes. So this was, I think, sort of a eureka moment after all of these previous failures to actually sort of see this infection coming up and showing that it had basically all the hallmarks of a chimpanzee infection or a human infection with hepatitis C virus. So that allowed us to actually begin to do genetics. And so we could, start with the transfected RNA clone sequence and, look at virus evolution and the host immune response in these animals. We could make mutations and see if these potential drug targets were in fact important for replication. And I guess this fulfilled Koch's postulates in terms of forcing this virus now through this interferon, through this artificial cDNA step, and still being able to sort of initiate a disease. So Koch's postulates fulfilled. And we had a functional, viable HCV RNA genome sequence. So you would think, being able to make, milligrams of this stuff, that you could begin to sort of identify, different cell types and conditions that allowed replication. And unfortunately, even though we had these sort of chimpanzee validated clones, that didn't turn out to be the case. So this is back in 1997, 99. So sort of 10 years later, we actually have a functional clone, but we still don't have a cell culture system. So the next landmark was actually from Volker Lohmann and Ralph Bartenschlager at the University of Mainz. And I think we were all kind of getting to the point where if we cannot solve this problem so that we can work with this virus in the laboratory, we're going to have to just move on and do other stuff. So Volker and Ralph sort of made what we call a subgenomic replicon construct, where they basically chopped out the region of the genome encoding the structural proteins, substituted that with a neomycin resistance gene, had another internal ribosome entry site to promote translation of what was believed at the time to be the non-structural proteins of the virus that were required for RNA amplification, and also keeping the 5' and 3' ends, as I've mentioned to you. So this worked, and they got sort of these very rare colonies that were, resistant to G418, a neomycin analog, and they had persistently replicating sort of HCV RNAs, really sort of quite amazing. And this was the first cell-based system that supported HCV replication in these human hepatoma cells, HU87s, and it allowed people to begin to develop candidase inhibitors and unbiased screens. The problem was that it was really inefficient, and there was only about 1 in a million transduced cells that actually initiated replication. So we reproduced that pretty quickly, and basically found that the colonies that were growing up harbored HCV RNA, but it wasn't the RNA that you started with. These RNAs actually had single point mutations, adaptive mutations that allowed these guys to sort of replicate in these HU87 cells. And a single one of these mutations could increase the efficiency of the system by more than 10,000-fold. And we could also cure cells that were growing up and find subclones of these hepatoma cells that were more permissive for HCV. So this, became a system where you could, develop reporter constructs. What one saw in the sort of drug development arena was that, sort of over the first sort of 10 years of HCV research, those of us that were actually tackling this, as sort of an academic lab, sort of felt that we were just going to get blown out of the water by, biotech and pharma. But a lot of these companies are kind of me too. And, until there was really sort of a cell-based system and they really started to crank, they weren't, I think, putting in as much effort to this as they could have. Anyway, so now we have RNAs that can replicate, we have adaptive mutations. Now can we complete the virus life cycle? So simple, you just take something that's not modified, put in the adaptive mutations. These guys could be transfected into these hepatoma cells. They, sort of replicated fine, no virus. So this is really getting pretty old. At least we had RNA replication, but no virus. So the next breakthrough in the field was from an investigator in Japan, Takashi Wakita. And hepatitis C is actually usually a subclinical infection, or, sort of fairly benign. But Takashi was looking at patients that had sort of rare fulminant disease from acute hepatitis C, thinking maybe that, the virus that was present in these individuals had a better chance of replicating in cell culture. So we all got kind of excited about this because when Takashi made a subgenomic replicon similar to the structure that I showed you for the Bartenschlager-Lohmann one, what he found was that could initiate replication in these cells without any adaptive mutations. And we thought that, well, maybe these adaptive mutations might be good for, chronic RNA amplification, but not so good for, sort of the later steps in the virus life cycle. And so this was called Japanese fulminant hepatitis 1. And I was lucky. I had I moved to Rockefeller at this point and had two fantastic postdocs, Brett Lindenbach, shown on the left, and Matt Evans, who actually may be joining you at a UC campus soon in the lab. And there were a number of other groups that sort of jumped on this and either, sort of looked at the JFH1, sort of full-length genome, or made chimeras where the structural region had been replaced with other subtypes. And basically it worked. So this could actually sort of generate a virus that was infectious in cell culture. It was infectious in chimps. You could recover the virus from the infected chimps and show that it initiated replication in cell culture. And then we could use these liver humanized mice. Basically, these are immunodeficient animals that have basically a liver that's repopulated with human hepatocytes. Basically a furry test tube for studying these hepatitis viruses. And we could actually, for the first time, sort of watch this virus spread. So this is a movie showing you accumulation of HCV-driven antigen on the left, and on the right, we've actually engineered this hepatome cell line so that if the hepatitis C serine protease is active, it basically cleaves off a reporter that's localized on mitochondria and then relocalizes to the nucleus. So for those of you that work on regular viruses, this probably seems like no big deal, but I mean, for us, it had been, sort of struggling with this for 20 years. It was significant. So I want to say a little bit about sort of the cure, and I'll try and move quickly here. And, when you think about hepatitis C, it does have some advantages in terms of thinking about that we could come up with a cure, and which is maybe a little bit presumptuous because I don't think there are too many chronic viral infections that have been cured, and probably this is the one. And, it's a cytoplasmic RNA virus. It doesn't integrate, so it doesn't behave like HPV. It doesn't have a cccDNA intermediate in the nucleus. It's stable like HPV. And we knew from the days of early treatment for hepatitis C that the virus is gone. So that if they're, basically virus free 6 months after cessation of therapy, which was driven by interferon, then the virus is gone, and accumulating evidence showed that if you get rid of the virus, you lower the risk of liver disease. So this is a snapshot of, sort of the evolution of treating non-A, non-B, and then HCV, which was, basically type 1 interferon, either higher doses, longer duration, pegylating it to stabilize it, or combining it with the nucleoside analog ribavirin. And in 2010, sort of about, roughly 50% of the people could be cured with this regimen. But, having high doses of systemic interferon is not a pleasant thing. And so people were nauseous. Ribavirin also causes hemolytic anemia. So a lot of people were, sort of not too happy about, sort of undergoing this therapy, even though it could be very important if their success rate wasn't going to be high and they had to suffer through all this. So the real drive in terms of improving treatment was to come up with an interferon-based therapy. And the three sort of winners of that, the viral targets that people arrived at, and I think the reason that these are viral targets in some respects has to do with basically the late timing with which we could actually, study HCV entry and assembly. So these were functions that were present in the subgenomic replicons that could be, assessed and targeted. So the serine protease that I mentioned before, the RNA-dependent RNA polymerase, and then NS5A, which is a multifunctional protein. It's not, as far as we know, an enzyme. It's involved in RNA replication and virus assembly. And it turns out, it was compounds that inhibit this was identified by basically a high throughput screen that was later refined. So pretty amazing. We now have these, fantastic treatments for treating interferon for 2 to 3 months. Hepatitis C is a very diverse virus, and it's estimated that in a single chronically infected individual, there are about 10 to the 12th variants being generated per day. And if you sort of multiply that by 50 to 100 million people that are chronically infected, that's a pretty huge, sort of virus variant space. And yet these combinations are very effective at sort of all of these genotypes with few exceptions. So in terms of what I've told you in this first part, although there's not a large part coming, is that, going from this mystery virus with a 5% cure rate driven by interferon in the 1970s to the identification Very quickly, diagnostics, at least in developed countries, were in place very quickly. So the blood supply is cleaned up. You won't get it. In 2011, the first of these direct-acting antivirals was combined with pegylated interferon and ribavirin, and it increased the virus cure rate, but it really had more severe side effects. And so a lot of patients were sort of waiting for 2014 and 2015, where we had these sort of interferon-free regimens. So there are some challenges in terms of determining who is infected. So actually, you would think that, you know, with these kinds of drugs, you should be able to just identify everybody and treat them and done. But it turns out to be, you know, involve, you know, issues of cost, challenging patient groups. So there was a high incidence of people that were infected with HCV and HIV that were difficult to treat, particularly in more advanced stages of liver disease. That has largely, I think, disappeared with these new regimens. So that's good news. Virus resistance hasn't been too much of a problem. And we still don't have a vaccine for hepatitis C that would be, you know, sort of a prophylactic vaccine. And part of the reason for that probably is that vaccine developers were sort of looking at the evolution of these antiviral drugs and saying, well, you know, if you can really cure this virus with a drug cocktail, is there really going to be a market, you know, for my vaccine? So, you know, we're actually not doing, you know, sort of all that great when you sort of look at how we've done. So this is really sort of an amazing paper published in the New England Journal a few years back that sort of looked at this from 2015 to 2020, you know, sort of in the first phase of when these drugs were available. And, you know, only about 11% of the people estimated to be infected with hepatitis C in the world have been cured. And if you look at the lower left here, you know, if you sort of look at the sort of rankings for most cases of HCV infection, which is sort of mimicked by the thickness of these bars. So, you know, China's at the top, Pakistan, India, blah, 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 Egypt, and the USA. And then sort of how they're doing, you know, sort of 5 years later, you can see that the US has actually moved up, you know, sort of in the hierarchy. Part of that has to do with what happened in Egypt. Sort of when you look at these sort of global numbers, and the Egyptians recognized many years ago that they had a severe problem with hepatitis C, with about 10% of the population chronically infected. And this was due to an anti-schistosomiasis campaign where the paraphernalia associated with administering this were not cleaned up in between individuals. And a lot of people got infected with genotype 4 HCV. And so in the case of Egypt, they recognized this as a huge problem, sort of negotiated to get sort of lower prices for the drugs that would be compatible with the sort of public health system in Egypt. And they managed now to cure more than 95% of the population with these drugs. So it can be done, you know, if you really sort of say you're going to do it and institute the infrastructure to do that. And we should be able to do that in the United States. So still no vaccine, as I mentioned, and we really don't understand sort of the disease progression of hepatitis C. And, you know, why that is important, I think, is that, you know, the good news is that if you clear HCV from the liver with these DAA drugs, that you do lower the risk of liver disease and accumulating evidence for cancer as well. The bad news is that even if you cure HCV and eliminate the viruses, and this is true also for suppressed HBV, that, you know, that's still a risk factor for developing cancer. So that people that are treated and successfully get rid of the virus, which is most of them that are complete the treatment regimen, you still have to be monitored for the development of liver cancer. And also, we don't really understand completely about sort of how this disease this liver pathology is generated. It's adaptive immune response probably dependent. And whether or not, you know, advanced fibrosis or cirrhosis, are these sort of reversible? Are there patient-specific differences in that? So we haven't really been able to figure that out. So one of the attempts to do this is to, well, I think we needed a better model. We can't use chimpanzees anymore for biomedical research. We tried, and others have tried, to adapt the virus to mouse cells. That didn't work very well. Genetic humanization, sort of define the human factors that are necessary for the human tropism of this virus, and then basically make a transgenic mouse that expresses those. We could get a transient infection with HCV in those engineered animals, but it didn't go chronic and it was very short-lived. Or you can look in nature and see if there's something similar out there that you could use as a model to study. So I was, very lucky to have Ian Lipkin and Amit Kapoor at the Mailman School of Public Health at Columbia, who sort of turned into being virus hunters, because with next generation sequencing, we can go out there and sample all these different sources, whether they be patient serum or animals or whatever, and see what's potentially there. And so every once in a while, Ian would call me up and say, "Hey, I found a new hepatitis C related virus in horses." So horses they're not really compatible with the Rockefeller campus. So we started working on them, but we had to sort of collaborate with the Cornell Ithaca. He then sort of called up and said, "Well, we've got this deer mouse that's infected with a virus." So we got deer mice, got the virus. Yes, they became chronically infected. We didn't see any evidence of disease. And then we found that, basically deer mice are phylogenetically so far away from laboratory mice that sort of all of the reagents and tricks that we have for working with laboratory mice would have to be reinvented in that species. So we tried putting the virus in highly immunodeficient laboratory mice and so on, nothing happened. So in the meantime, things are marching on. People are finding hepatitis C related viruses in bats and gibbets. But sort of the real breakthrough for us was basically when Ian teamed up with a couple of pest control agencies in New York City. And he trapped a bunch of these Norway rats. And, there were two lineages of hepatitis C related viruses in these rats that had been scurrying around right under our noses the whole bloody time we were trying to develop an animal model for this guy. And so anyway, we got the virus. And basically this Norway rat Hepatitis can be transmitted to laboratory mice. The infection is very similar to what we see in HCV infected people. I'm not going to sort of go through all of these things because I think I'm running low on time, but we think it's a good model for hepatitis C associated infection and disease. And you can actually get these animals which are naturally able to resolve the infection in 2 to 3 weeks. If you transiently deplete CD4s before you infect them, they become chronically infected and they develop progressive liver disease, as I'll show you. And that's true even after the CD4s come back. So, so now we have, a chronic infection model. So I guess one of the things that we were interested in is, sort of what's the pathology associated, with these models. And I guess the thing that was interesting was that we could show during the progressive chronic infection that you can, basically generate a disease state where you get progressive fibrosis. And that old mouse that I showed you on the previous slide there, when we had somehow escaped euthanasia in the animal house, and when we sacrificed that animal, it had this terrible looking liver. But, we didn't know it's an old mouse, and sometimes, sort of old mice have terrible looking livers. So we sort of set up, sort of a study to sort of assess the impact of chronic infection in this model. And that study was interrupted by COVID-19 and we weren't able to get all of the samples that we wanted to from these animals. But what we could show is that, they sort of developed, progressive sort of fibrosis and inflammation, as read by Neil, a pathologist. And you could see that was very evident to be, sort of infection dependent at these 7 and 12-month time points. At 18 months, sort of the livers of even the uninfected mice are not looking that good. So if you look at the transcriptome, there are a lot of similarities in the sort of liver transcriptome of these animals and chronic HCV. Won't go through the details. And then there was this surprising observation. So these are sort of mock infected livers shown here. And these are ones from the chronic NRHV infection. And basically you can see these tumors sort of showing up on all 4 of these livers, which we are sort of currently sort of characterizing further. And the frequency is really high. Almost all of the males get what looks to be something very similar to hepatocellular carcinoma. And about half the females end up with this at this 18-month time point. And that's similar to what we see in chronic hepatitis C in people. So some of these have driver mutations. Some of these tumors have driver mutations, some of them don't. And so we're pretty excited about this model if we can actually succeed in getting funding to pursue it. So these are some of the things: oncogenesis, sex, and comorbidities that accelerate sort of liver damage and potential disease state. The role of fibrosis, if you treat these animals with things that are anti-fibrotic, so things like targeting senescent cells, for example, does that sort of interfere with the development of HCC? And we now have conditions where we can actually cure these animals at will. So we can allow them to be chronically infected for different periods of time, get rid of the virus, and ask, do they still go on to develop cancer? Kind of something that is important in terms of thinking about our chronic HCV patients that have been successfully, sort of virus cured at different times in pathogenesis. Immunology and host genetics. It turns out, for those of you that are familiar with the collaborative cross, this is a collection of mouse strains that are very different from each other, but are inbred. So you can use these to interrogate the phenotype of whatever you're interested in, whether it's, fatty liver or, hepatic infection. And we found one of these, Tim Sheehan at UNC, which becomes chronically infected without depletion of CD4 T cells. So we can cross those to C57 black 6 mice and sort of map the determinants that are important for that. So I won't go through this. These are some of the other things that are going on in the lab right now that are, I focused on things that are HCV related. The Norway rat hepatovirus is actually the last remnant of HCV work in the lab, but there's a lot to do. And so this is an acknowledgement slide for the people that were involved in this sort of last work that I told you about that'll be published soon in the Journal of Hepatology. And you can see our canine support staff here in the front, our Australian shepherds. And so the HCV success story, a long road, really involving so many people, lots of basic science, great work from biotech and pharma, the clinicians. It really was, something that involved thousands of people, and I hope we get to see it to its inclusion. And I thought, just because of the time that we're in now, sort of this Lewis Thomas quote is something that I really enjoy. In the course of scientific progress, things tend to pop up. Most of the celebrated achievements had their origin in moments of surprise in the laboratory, unplanned for, unanticipated, unpredicted. No committee convened to survey the future prospects for biomedical research could possibly have guessed at the things that lay ahead. No branch of government could have laid out detailed plans for the scientific needs of the future beyond asserting that what the country needed was more fundamental knowledge about the human form and function, about the agents and influences responsible for disease, and in a certain sense about nature itself. Scientific research works. It is the only way to get at the underlying mechanisms of disease and the only way to learn what to do about them. This is now under threat. I hope that that doesn't continue. And so a few final Rudi thoughts. One's career is often determined by chance encounters, and I think that's probably something that we could all attest to. Who you encounter makes a big difference. Persistence and rigor does pay off. sometimes. And supporting fundamental curiosity-driven research is key, and a bright future lies ahead, we hope. So I'll stop. Thank you.
Thank you. That was really, really inspiring story. We'll take a couple of minutes for questions. We're a little bit over, but seems like a good time to do that. Let me just start with one in terms of lessons. Obviously, the resilience is key. When you counsel young scientists and they look at you and say, "This is the career I want to have," any other sort of advice you give them that you think they need to know starting out?
Well, I don't know. I think you have to, sort of follow your curiosity. And I hope that we have a system that will, sort of allow people to continue to do that. So I find myself really, sort of privileged in the sense that somehow I managed to sort of exist, sort of doing my hobby for all these years and actually getting paid for it. So yeah, I think, you got to be curious because things don't always work in science. So you've got to be ready to come back in the next day, roll up your sleeves and, test your next thought.
Right. Don't always listen to your mother. Yeah, go ahead. Say who you are.
Thoughts on the vaccine now that the antivirals have not really done their job. So do you think that there is a chance, or is the virus?
Well, I guess the question was, what about the vaccine now that the antivirals haven't done a job? I think a lot of the problems with the antivirals is administering them and identifying, sort of the sort of high risk people and, getting them into treatment. And, with vaccines, you face, maybe now more than ever, the same kinds of issues in terms of implementation. But I hope that, again, messenger RNA, lipid nanoparticles, all that stuff, sort of aside, which it seems very, sort of promising to me, that I think there should be some rekindled interest in generating an HCV vaccine. So there are groups that are, working on this, maybe not at the scale that I'd hope. I sort of thought, well, sort of when, the capitalization of Moderna was through the roof, that, maybe they could carve out 0.01% or something like that to develop a hepatitis C vaccine. That doesn't seem to have happened. So I think there's enthusiasm and there is a need. And I think it's possible. I think it's just a matter of, testing things. It's come to the point where, and this is kind of interesting, that the treatments are so effective that we're discussing the possibility of having human challenge studies, because the actual cure rate is more like 99% and probably, sort of even higher than that in a sort of the more acute, sort of stage of infection where you would be testing immunogenicity and resistance to chronic infection.
Take one more. Yeah. Bye.
Thank you for your talk. Lucy Williams, social worker with hemophilia. A lot of our patient population had HIV, hepatitis C. Question, do you see treatment expanding to later stages of liver disease for hepatitis? Or?
Oh, yeah, I think, certainly people with later stage should be treated. It was all, I think in some venues, when these drugs became available, people were interested in, sort of treating the people with more advanced, liver disease first because they had a higher risk of dying. But I think, obviously sooner is better. People that have an acute resolved HCV infection are not at increased risk of liver disease. And so I think, well, my dream would be to just treat everybody.
Just piggyback on that last one. That's any lessons for HIV cure and HIV treatment from what you've told us?
Well, that's a hard one, and I don't know if cure is going to be achievable for HIV. I don't know. Certainly, we are seeing some promising results with sort of persistent control. That is sort of lasting and regimens that don't require frequent administration and, sort of provide suppression. But, Melanie would be the one to sort of answer this question. People go back and forth. I don't think we should stop trying. That's the bottom line. And I think this is the same for hepatitis B.
Thank you so much. It was a really inspiring talk, inspiring career. Thanks. Great. Appreciate it.